Hey, everybody. Welcome to Thought Leader Thursday. I'm Beth Usry. I'm the Director of Customer Engagement here at Quality Executive Partners, and today I'm going to be the moderator of the webinar. This webinar series is brought to you by Quality Executive Partners. For those of you who aren't familiar with us, we are both a consulting and a technology firm that provides innovative solutions for the life sciences industry. In 2019, we launched Virtuosi, and it is literally transforming learning and education in the pharmaceutical industry. Our first series combines video and VR immersives in an 80-hour curriculum focused on aseptic manufacturing and microbiology. And I have another very exciting announcement to make today. I'm thrilled to introduce Takati, the next evolution in education brought to you by QXP, which will be launching in 2021. Takati offers an advanced level of learning experiences for site and corporate leadership on complex issues facing the industry. Takati includes interactive lessons, discussions, and workshops with SMEs and peers to provide a deployable, practical knowledge. Our first session will be on advanced forensics auditing for data integrity, and it's scheduled for February 1st through the 4th. Our very own executive leadership team members, Crystal Mersch and Brian Duncan, will be leading this session. Each session is limited to 48 participants, so you're going to want to sign up to that early. Registration and more information is coming soon, so check us out on LinkedIn or email us at takati@qualityexecutivepartners.com. On LinkedIn, you can follow us at Takati, and that's spelled T-O-C-C-A-T-I. So we look forward to followers and look forward to additional information on the Takati program. So it's an absolute pleasure to lead this webinar and welcome back those viewers who have been able to join us the past couple of weeks. This is the third installment of 4 of this webinar series, and we'll be having 2 hosts again this week. And I'm happy to start by welcoming back Vanessa Figueroa. Vanessa earned her BS from State University of New York and her master's from San Jose State University. Vanessa has more than 15 years of experience in industry, and she has worked in the laboratory and manufacturing areas. Vanessa is a published member of PDA and an active contributor to the American Society of Microbiology. And if that isn't impressive enough, Vanessa is one of our lead developers for Virtuosi, and not only creating the technical content, but also presenting the content as our on-screen talent. So let's give a warm welcome to Vanessa Figueroa, our Executive Director of Microbiology.
Hello, Beth. Good morning to you. Good day to everyone. Thanks for having me back, and really excited to talk about bacterial endotoxin today.
Yeah, thanks, Vanessa. So great to see you, and thank you so much for joining us today. Our next panelist today, I'm also happy to welcome back Tyler DeWitt, who you may recognize as our other on-screen talent for Virtuosi. Tyler earned his BS from Brown University and his PhD from MIT. Tyler's scientific background is in microbiology and biochemistry. A lot of his work has been focused on bringing new technologies to teach cutting-edge science, particularly in the pharmaceutical industry. Tyler is also one of our lead developers of Virtuosi. Let's welcome Tyler DeWitt, our Executive Director of Innovation.
Hey, how's it going?
Hey, so good to see you. So good to see you. You know, giving all of you access to people like Vanessa and Tyler is exactly why we started Thought Leader Thursdays. I think that allowing our experts to provide answers to your questions and information and knowledge on specific topics has been super, super helpful. So now that we've introduced the thought leaders, we'd love to know more about our audience. So we have a polling question to get us started. So for those of you who haven't joined us before, this is the first of 2 polls that we're going to have today. And this is a great chance for you to warm up on answering and be an active audience member. So you can see the question has come up already. Which method do you use for bacterial endotoxin testing of product? So go ahead and answer the question there. I'm going to take a look and see how it's going.
Oh, it looks like a lot of folks are using JAVPlot, and then maybe a combination.
Yeah, the answers are still coming in. If you haven't answered yet, go ahead and answer. I see still coming in. We'll give it just a few more seconds here. If you haven't answered the poll question, go ahead and do so. All right, I think we've got just about everybody responding, so great. Let's take a look and see. Let's see what people responded.
That's great. Yeah. So we've got 38% using gel clot technique and 38% using more than one or a combination of the above. That's great. And folks representing 8% don't do VET. So it looks like, the other 92% of the audience today does do VET. So this is going to be very relevant for them, and it's exciting. Thanks for that.
Great. Yeah, so the polls are just one way that we are going to have active participation during the webinar today. So one of the most popular parts of our webinar format, and the thing that I like the most about QXP's webinar format, is the question and answer session. So you all get an opportunity to ask Vanessa and Tyler any questions that you have on bacterial endotoxin testing or on Virtuosi, and enjoy the benefit of their expertise in responding to things that are happening in your day-to-day work. So you can submit a question anytime during the webinar. So you don't have to wait until the question and answer section at the end. So anytime throughout the webinar, you can feel free to ask a question. You're just going to go to your control panel in the upper right corner of your screen, click on the orange arrow to expand the menu box, select the questions box, type your questions in, and you could submit them right there. And we look forward to seeing all the fantastic questions that are going to come in. One last technical update before we get started. If you're using computer audio and you lose audio at any time during this webinar, you can easily switch to phone audio by clicking on the phone call button in the audio box. So the call-in numbers will appear when you click phone, and then you can dial into the webinar that way. So if you lose audio, there's an option to call in. Okay, well, without further ado, why don't we go ahead and get started on today's topic of bacterial endotoxin testing. Today, we'll get a chance to see educational videos that help us with basic bacterial endotoxin methods and depyrogenation. You will also get to see a demonstration showing proper techniques for lysate sensitivity testing in our virtual laboratory environment. Please notice that we go in and out of videos and we come back to presenting. You may experience a short delay, or your screen may briefly change color, so just bear with it. It's just switching back and forth. And also, if you're on a mobile device, you may need to tap or swipe the screen as the video loads, and you may need to adjust your volume as well. So if you're having issues, you can use those cues. So before we get focused on how of endotoxin testing, let's talk about why it's so important that products don't contain endotoxin.
Yeah, thanks, Beth. So at QXP and Virtuosi, we really like to start with the why, right? We find a lot of our clients, and moving in a fast-paced world in 2020, we're always focused on the doing and the how and executing SOPs, right? But we'd like to level set both with our audience, with the learners, and with our projects and folks that we work with on the consulting side with the why. And testing for bacterial endotoxin is one of those fundamental product release test requirements for a batch purporting to be sterile, right? The industry really relies on BET and sterility testing for that finished product release in microbiology. BET is also used to test ingredients, such as what are for injection or raw materials that are part of that formulation for a sterile product. And if you understand, bacterial endotoxin itself is the chemical compound that's part of the cell membrane, right, of a gram-negative bacteria. And it can be very, very harmful when found in injectable products. It causes, a pyrogenic response, right, a fever. And so the last micro webinar that we had 2 weeks prior on sterility testing drove home the same point. And it has to do with sterile products in general, that they must not contain any microorganisms. And in this case, for BET, they also must not contain any remnants of those organisms either. And that's the key point we'll talk about today.
Okay, great. Thanks, Vanessa. So both Vanessa and Tyler, you both know me, and you know that I'm a biologist, not a microbiologist. But from what I do understand, there's multiple test methods for detecting endotoxins, right?
Right. As Vanessa mentioned, right, endotoxin is this molecule that gets into our blood, causes a big immune response, inflammation, but particularly fever. And so endotoxin was initially tested for in rabbits. You'd inject the sample into rabbits, and literally, if your experimental rabbits got a fever compared to the control, well, you'd know there's some endotoxin in that sample. This is laborious. It's a difficult test to do. It's still done occasionally, but generally the standard has moved to sort of more traditional laboratory analytical tests, which tend to be based on a lysate that clots or causes a chemical change in the presence of endotoxin. So the simplest of these is what we call the gel clot test. And then there are more advanced methods that tend to be automated with names like chromogenic and turbidimetric methods. And we actually have a good sort of introductory clip from Virtuosi that talks a little bit about these different methods and gives some more background specifics. This would probably be a good time to take a look at that so we can all sort of orient ourselves to the basics of these methods.
All right, that sounds great. Let's go ahead and take a look.
The bacterial endotoxin test can be carried out in a qualitative, semi-quantitative, or quantitative manner based on whichever one is necessary. The qualitative analysis via the gel clot method is the simplest and least expensive, as it does not require any specialized equipment. The gel clot method can also be conducted semi-quantitatively when a sample titration, which is a series of increasingly dilute samples, is compared to a standard curve. However, in either case, the gel clot method relies on training and careful observation and can be subjective. Other methods can be more expensive, but they offer very precise results, either at a specific endpoint or based upon a rate of change, the so-called kinetic methods. The chromogenic method is a type of photometric measurement of color change, which uses an instrument to measure the appearance of color of an enzymatic reaction. The endpoint chromogenic assay is defined when the period of incubation ends and a chromophore is released with the addition of an enzyme reaction terminating agent. Whereas the kinetic chromogenic assay is defined using the rate or the time needed to achieve a specific color development throughout the assay. Turbidimetric methods measure the turbidity or cloudiness of a substance using a photometer. The turbidimetric method is perhaps the most commonly used method for endotoxin detection. The endpoint turbidimetric assay is defined as the relationship between the concentration of endotoxin and the turbidity of the sample after the incubation period, where the kinetic turbidimetric assay is defined as the time needed to develop turbidity or the rate of change of the reaction mixture. These methods will be discussed in greater detail throughout this episode.
That was really fantastic. So the laboratory methods all use lysate, right? So isn't that a reagent that comes from horseshoe crabs? Did I hear that somewhere?
You did hear that, and that's correct. We call it lysate for short, or LAL, Limulus amoebocyte lysate. Yeah, it's literally extracted from the blood of the horseshoe crab, which is this kind of weird looking blue blood. And what's interesting about it is in the mid 1950s, some researchers at Johns Hopkins discovered that the horseshoe crab blood would clot in the presence of endotoxin. And what was cool is they weren't looking to discover a new endotoxin test method. They were just literally looking at how the horseshoe crab could protect itself from bacterial infection. And they ended up stumbling upon this thing that, really revolutionized pharmaceutical testing. But so anyway, so lysate, the lysate that we use is an extract of the blood components from the horseshoe crab, and it has all the enzymes in it that detect the endotoxin and then cause the clotting. So this lysate really serves as the basis of all the different types of endotoxin test methods that we use in the lab.
That's really, really cool. So did they actually have to hurt the crab to get their blood?
Well, getting the blood from the horseshoe crabs is a little bit like a Red Cross blood donation. The crabs are captured, some of their blood is extracted, and then they're released back into the wild. So they make it. But I will say it's somewhat of a controversial practice, and there are lots of conservation efforts around horseshoe crabs. the industry is just starting to embrace some replacements for lysate that are produced synthetically in the laboratory. Recombinant factor C is the synthetic replacement for LAL, and we can talk about this a little bit later, but it's sort of a slow industry movement towards the embrace of some of these synthetic replacements.
Okay, good. Thanks for explaining that. I was worried about the horseshoe crabs. Okay, so now that we have our lysate, now we're ready to run the BET. Yes?
Not quite. One of the things that we need to do is we need to confirm the sensitivity of the lysate to essentially ensure that the lysate will clot, that it will react within an appropriate concentration of endotoxin. And this is really important to ensure that our test is valid. So with a gel clot, this procedure of confirming lysate sensitivity is done manually. With more advanced automated methods, it's sort of done automatically. But we wanted to talk about how this is done manually with the gel clot method. We have a clip from the Virtuosi VR immersive experience on this that we want to share in a minute. That shows some of the aspects of confirming lysate sensitivity with the gel clot assay. And when Vanessa and I and others were sitting down to sort of scope out what the VR immersives would look like during the development of Virtuosi, we decided that we really wanted to put in lysate sensitivity confirmation with the gel clot method, even for folks who don't use a gel clot method. Because if you understand how this technique is done manually, if you understand how it's done, if you understand the decisions that need to be made, then you'll have the foundational understanding for being able to see how this is done in any of the more automated methods. So we have a very why-focused process in teaching. We're always focused on the basics. And I think this shows really nicely in this immersive. So we can queue this up. And we sort of start in the middle of this immersive. It's sort of a long procedure, so we want to show you a few highlights from this. And I think Vanessa will start off talking us through a little bit about what we're seeing in this VR immersive.
Okay, cool. We're ready to go, so we can go ahead and take a look. Let's get the video queued up.
Yeah, so hello everyone. So we start off essentially, as Tyler said, in the middle of our procedure within Virtuosi in the VR lab. And this is after preparation of the standard dilution series has been completed. So the demo is about to show you how the lysate is automatically reconstituted in VR. And so what he's doing is he's looking at the SOP on the wall and he clicks the reconstitute the lysate button. So this is filled with the LAL reagent water. And then you can see the manual gentle swirling for about 30 seconds. And that's part of the reconstitution process of lysate. From here, you see them pick up the pipette aid. He's given a warning to ensure that the tip box is closed while he's making other movements. He grabs a sterile tip and he's going to dispense 100 microliters into each of his reaction tubes, starting at the lowest concentration, so the 0.25 lambda. Then he's going to put that on the vortexer. It's cool to hear that vortexing sound in VR. And really, this is meant to show you that you have the supplies, the pipettes, the pipette aid, the tips, the lysate, and the ability to vortex inside a virtual laboratory space. And you even hear the clinking to make sure that that final dispensing is happening, ejection at the end of the tip. You've got the clock there to show how you're vortexing. And then this will give you a quick highlight and Tyler will take it over from here.
Yeah, and so what we're seeing here is sort of the interpretation of the results and the recording of the data. For anyone who's done the gel clot method before, you know that you take these final tubes and you invert them nice and smoothly 180 degrees, and you essentially assess whether or not a clot has formed in that tube. And for each of the replicates and for each of the different concentrations of endotoxin, we mark whether that corresponding tube has a clot or doesn't. And so that's what the pluses and minuses here are that the user is selecting. Now, those different values go into the calculation that we do to determine what the lysate sensitivity is. You can see the calculation up there on the wall. Those numbers all came from the different values that we indicated in that sort of data reporting chart in front of us. We'll do the calculation, and we'll be able to determine whether or not that lysate sensitivity is within the appropriate range, and essentially whether our test is valid.
That was really, really informative. I've been in VR before, and I do understand just to that we're just seeing a 2D view of inside the laboratory. So once you're in there, you're able to see 360-degree view, right?
Yeah, you can turn your head all the way around. You can walk around the lab if you want, our virtual microbiology lab. In specifically BET, there's different user interfaces. So you've got the SOP checklist on the wall in front of you. You've got all of your lab supplies there on the lab bench in front of you. And then you've got the ability to interact with different parts. We also show you how to do pipetting in this interactive experience. And so you really get to practice all the necessary steps in order to do and execute the gel clot technique for bacterial endotoxin. Every time I go in seeing these VR clips, I feel like I'm there and I want to stay there for way much longer, right? So unfortunately in these webinars, we can only show you a couple minutes to let folks sort of orient themselves to see what's possible in VR, right? The microbiology lab, what it looks like, the 3D rendering of this space is just so exquisite. And just how smooth and normal, you really feel like you're in this space. You really feel like you're picking up a pipette aid. And what's really cool is, if I could, I have one of the controllers here connected. You use the back of the controller trigger as if it's the pipette aid itself. I mean, this is just like revolutionary. It's really, you feel like you're there. You feel like you can practice. You're in a safe space. And so VR is definitely truly amazing. It allows you to practice the actual techniques you need to make those mistakes, right? Pipetting, vortexing, adding the reagents, making sure that they're in the right sequence. It's tracking each of your dilutions. It's tracking whether or not you added lysate to that reaction tube. And if you didn't, your results will be skewed at the end, and you'll have to do it all over. But these real instruments and supplies don't cost anything, right? They're in a virtual setting, and it's important for you to have that space to be able to practice and learn these techniques and hone those skills as a microbiologist. So it's a lot of fun. It always excites me to be inside VR.
Yeah, that's the part that I love about it, that you could get in there and practice and not be worried about making mistakes, not be worried that someone's standing over your shoulder and you're nervous if it's the first time you're doing it or the 100th time you're doing it. It's still, you can practice in there and a risk-free environment to make mistakes in. That's kind of the point. We learn best when we're learning from our mistakes. So I love that. And yeah, the sound effects, like, so cool. So. Okay, enough about the cool VR aspects. So now that we have completed the sensitivity testing on the lysate, now we're ready to test samples, right?
Yeah, I love your biologist eager perspective. Yes and no. So it depends on where in your routine process you are. In theory, you still have to perform method suitability testing, right? In order to confirm validity of the test during method suitability, you need to consider a couple things. First, you'll need to determine how much of the sample that you can dilute in order to give you still valid results, right? That's what we call the maximum valid dilution. Then you need to determine the absence of inhibition or enhancement effects, right, on the measurement, your ability to measure that endotoxin in the sample. This confirms that the composition of the sample doesn't interfere with your ability of the lysate to react. So if it's a gel clot method, either by clot, if it's the chromogenic, whether it's a chromophore color change, or the turbidimetric, the ability for it to render turbidity. So once these are established for your product, then you're ready to actually test the samples in routine. And it's really key that you understand every manufacturer makes lysate, right, from their natural sources. And so you essentially have to calibrate that lysate with your endotoxin every time you receive a new lot of material. But you don't have to re-execute the whole method suitability. You just have to determine what is appropriate for your product samples at the time of method suitability. So I wanted to make sure we covered all aspects of that, and not just like, hey, all I need is lysate and I can just pull some endotoxin from the shelf and go. Right? There are a lot of things and aspects of the program you need to consider first, especially unique to your product.
Yeah. So I just had a thought. So we're going through all this trouble and doing all of this work to determine if there's endotoxin in our samples, but how do we know that the materials in the lab or there's not something else that's contaminating our samples with endotoxin?
Yeah, I mean, that's a great question. And it's also essentially part of method suitability, right? So determining the supplies, the materials, really what's going to, what are the conditions that you're actually considering during method suitability. We think about these things up front. So you have to think about the pipette tips, the reaction tubes, all of the materials that you're going to use in routine for your bacterial endotoxin test. And these are evaluated for whether or not they are as low to no pyrogen as possible. In order to render these things pyrogen-free, we use the process of depyrogenation. So it's got that depyrogenation word in it. And this is also, like I said, evaluated during method suitability. So it's on you guys, and this is really in real life experience, this is one of the things that the agency, regulatory agencies look at. They look at your materials to make sure that they're not plastic or they're not somehow either contributing or helping to promote the binding of endotoxin to the walls of a reaction tube or a pipette tip. So they look to make sure that you have given thought to these materials and how they are low to no pyrogen. But I think that we have the digital version of Tyler in Virtuosi, who will take us through the concept of depyrogenation. And maybe we cue up that clip now and he can tell us a little bit more about that.
All right, great. Let's take a look.
A key part of this control is the removal of pyrogens, or the depyrogenation of materials, components, and equipment that are used in the manufacturing process. Remember, too, that materials used in the testing of pharmaceutical products should also be depyrogenated to ensure that they'll not interfere with the ability to accurately measure endotoxin in samples. It's important to reinforce that depyrogenation is different from sterilization. Sterilization is the destruction or removal of all living microorganisms. However, most sterilization processes do not remove endotoxin because it's such a robust compound and remains even after microorganisms are killed. Instead, there are some specific methods that can reduce or remove endotoxin from materials, components, and equipment. Depyrogenation is the removal or destruction of pyrogens, and particularly endotoxin. Endotoxin removal from an article is validated by showing a multi-log reduction in the endotoxin load from an initial concentration. Dry heat depyrogenation usually involves exposure to hot air at 250 degrees Celsius for a minimum of 30 minutes. Since depyrogenation cycles are run at much hotter temperatures than sterilization cycles, a validated depyrogenation cycle will also effectively sterilize. But it's important to remember that the reverse is not true. A sterilization cycle will not necessarily depyrogenate because endotoxin is thermostable. A dry heat depyrogenation process should start with materials that already have a low pyro burden. Pyroburden means the total amount or burden of pyrogens that's present in a material or sample. Rinsing with high purity water can sometimes help to reduce the endotoxin to a low starting level. For some materials, water rinsing may be all that's needed. Yet other materials are exposed to high temperatures during the annealing or extrusion processing, which will reduce the amount of endotoxin. Regardless of the method chosen for depyrogenation, the success of the process must be proven with a high degree of confidence.
It was so fun to see you guys on screen and have you live with us. There's Vanessa, there she's back. To have a deeper dive on these topics. I'm so excited about it. So
I feel like Tyler and I are competing about with our hair days. Like I feel like I had a way better hair day there, and your hair was so perfect, Tyler.
You guys look fantastic all the time.
There you go.
So I think this is a great topic for us to pull the audience and hear from the participants to see how they're performing endotoxin testing before we get into the QA section. So you may have gone to full screen to watch the video, so you may have to come out of that mode to answer the poll. So here we go. Which parts of bacterial endotoxin testing are you responsible for at your company? So go ahead and submit a response there. I see the responses coming in. Keep it going here. I see lots of people answering. And we've still got some trickling in. We'll give it just a few more seconds here. All right, a couple more trickle in. If you haven't answered the poll question, go ahead and do so now. And we're going to close it out in about 5 seconds. All right, it looks like everybody's answered. So let's go ahead and close the poll and display the results.
Good mixture, a good mixture. So you see, we've got folks responsible for implementing the program. We've got a lot of folks who do the routine testing, about 25%. Another 25% for those who review test results and investigations, right? So that blend is common to see. And then we've got 40% with none of the above, which means that they're on this webinar to learn something new, and that's always exciting, and it's great to be able to connect with those folks as well.
Yeah, that's fantastic. It sounds like there's a really good mix there in our audience. So Vanessa, Tyler, thank you so much for walking us through the basics of bacterial endotoxin testing. This is a great time to open it up to questions from the audience. If you haven't asked a question and you would like to, you can go into the question section in your menu bar and go ahead and submit one there. We've already received a bunch of questions here. So why don't we go ahead and get started? I'll pull up the first one. Says, hey there, I'm a non-microbiologist. I can relate and know this might be basic, but I've heard that there's a potential problem with a COVID vaccine in horseshoe crabs. What's going on there? Are you guys up for talking about COVID?
Yeah, I can start out, and then I'm sure this will be one of those things that Vanessa and I both have something to say about. First of all, we don't discriminate against non-microbiologists here, and it's cool to have people with a diversity of backgrounds asking questions, and that's always fun to talk about things at multiple levels. So all these COVID vaccines that we're talking about, we're going to be trying to push out a lot of doses very, very quickly, and obviously this is going to be a parenteral product that needs to be free of endotoxin. So how is all of it going to be tested? As we said earlier, primarily right now, endotoxin testing is done using the lysate derived from the blood of the horseshoe crab. And so there has been some reporting in the press about potential shortages of that lysate, the tremendous amount that we're going to need to ensure that all of these vaccine doses are pyrogen free. And so that sort of leads into what we were talking about earlier with some of these synthetic alternatives, recombinant factor C, which the pharmaceutical industry has been a little bit slow to embrace. So there are lots of concerns with horseshoe crab population and conservation. You know, will the populations be able to withstand sort of this large demand for lysate? How about, you know, the validation of using these recombinant proteins and so forth? And so it'll be really interesting to see sort of how these different factors all work themselves out as we sort of work towards this unprecedented challenge that we've never had before. I'm sure Vanessa's got some stuff to add as well.
I mean, I think that just doing research, I think that the what's being told to the public and the press is sort of a mixed bag, right? We don't know exactly how many companies are going to actually go straight to production, right? It depends. There are a number of companies, both in Europe and the US and around the world, that are in phase 2 and 3 clinical trials that are close, that are gearing up for production to be staged, right? And then I know just in America, we need billions of doses, right? And so this does have an impact on the industry, but then I also read from some of the vendors and suppliers of lysate who work with these horseshoe crabs who produce and manufacture the lysate that we are all using, that the supply is not a concern, and that, you know, it's sort of something we're going to take one day at a time. So there is, you know, sort of mixed reporting on it, and neither of us work for either of those companies, whether you're on the vaccine production end or you're on the lysate production end. And so it's really hard. We're just as informed as the public itself, and you know, really it's just staying tuned to understand. But I think that the message is, at least for me and for this webinar, is the relevancy and the importance of this test method, right? And you can never lose sight of something. We talk about bacterial endotoxin and sterility testing as, like, you know, the grandfathers of the finished product release testing requirements, right? They are something that every pharma microbiology lab does, and it's required to release the batch. And so it's easy for a company or for a lab to get complacent in the processes and the execution of the routine testing and stuff like that. But we here are always here to remind you that it's important to understand the fundamentals, the basics, understand why, and be able to trust, you know, your skills and being able to consistently, reliably test this product. And I think, you know, it's great for microbiology labs. We're going to be testing a lot of bacterial endotoxin samples for all these COVID vaccines, and it just shows how important the role a microbiologist plays in the overall health of both the public and the industry, and being able to push out product that meets these standards of quality. So kind of full circle there, back to why micro and you know, what we do for BET is so critical to the industry.
Yeah, absolutely. I mean, just hearing you guys talk about topics that are happening to all of us today, and understanding the role of a microbiologist in the world for this situation that we're experiencing is it's pretty cool to be on the front lines of that. So
yeah, absolutely.
All right, next question. Do you recommend reverse or forward mode pipetting for BET testing?
Good question. Good question. So we actually endorsed forward pipetting inside Virtuosi specifically, and even in this specific, you didn't get a chance to see it here, but even in this specific interactive experience, on the wall to the left, there's a diagram of the forward pipetting sequence, and getting folks to understand how to use those skills is part of a different interactive experience. And so the structure of Virtuosi works such that we build on those skills, right? There are some, there's 3 tiers to Virtuosi, and in the basic level 100, you've got some of those basic techniques, and Tyler probably can talk more about this. He's more of an expert, and he wrote the content for that. But pipetting is an interactive experience that we have on the topic all by itself. And we teach you the basics of pipetting. So what it means to do reverse and forward pipetting, and why we think forward pipetting is important, right? What is a resting position? What's the first stop? What is the true eject mean? And why you can't, backwards pipette, and you don't want to, withdraw once you've entered into a solution. What are the consequences of those? What are the consequences of having residual, liquid on the tip of your pipette as you're pulling out? So there's a lot to consider, and I think we at QXP endorse forward pipetting. I don't know, Tyler, if you have any other thoughts to add.
Yeah, I think forward pipetting, in my opinion, is usually the way to go. It's generally simpler. It reduces opportunities for error, with the one exception that reverse pipetting can be really useful when you're working with an extremely viscous liquid. Now LAL, lysate is a little viscous, but that's not the level of viscosity that we're talking about here. Something where you're have a 50% glycerol solution, or something that's really, really, really viscous gets stuck at the end of the pipette tip. Now that's a situation where you might want to use reverse pipetting and so forth, subject to all the method suitability and so forth that you do. But generally, forward is going to be easier. It's going to be simpler for analysts to master, and it's usually the technique to rest on.
All right, great. Thanks for answering that question for us. Next one coming in, and this is also related to pipettes. We have a lot of pipette errors. Would you recommend? Do you have any recommendations that would help us with this?
Sorry, my audio is on mute. Yeah, I mean, the recommendations start with that core education. So obviously Virtuosi can help you with that. We have all the content on pipetting, and then you get to practice it in VR, which is a really great skill. But that doesn't replace actually learning and practicing it in real life with real instruments, right, with the real pipette aid. And so we cannot overstate the value of strong pipetting skills. And so just the awareness of having pipetting errors or sloppiness, or maybe attention, lack of detail to that process, helps you to informs you as either a lab supervisor or manager to know that, hey, maybe I need to take a step back. These folks need additional education. You can put together content and sort of structure delivery of practicing that in the lab. So get folks to understand there's a specific technique, there's a way to execute that technique, and here are the consequences when you don't take your time, when you don't understand the settings of a pipette, when you don't understand the types of pipette tips or pipettes themselves that you need to use, why it's important to use smaller volumes for more precise measurements, and why you can, when it's appropriate to use larger pipettes with a larger volume, say 9 milliliters, and what's the differences. You've really got to build that core knowledge and get folks really invested in understanding some of that core knowledge and the why behind each of the moments or the opportunities for error in pipetting. Because from the start to the finish of pipetting something, there's about 100 different opportunities for error. And all of those matter. And especially when you're pipetting very, very small quantities, the errors are compounded, right? And so understanding and educating the lab, taking them back to the basics, don't focus on real samples, don't focus on that. Have them start pipetting water. Have them start pipetting glycerol or other trickier substances. Have them pipette small quantities, large quantities, use multi-pipetters, use repeaters, and understand, what are the differences between all of these equipment, all these measurements, and why and how they relate to what you're doing in your lab and your company every day. So I don't know if you're pipetting organisms, or if you're pipetting water, or if you're pipetting for bacterial endotoxin, it doesn't matter, but attention to detail. It also takes a very specific person to appreciate the nuance and the potential for errors in pipetting, right? Attention to detail is definitely a skill that this type of person needs. And so taking it back to that, developing that core education and maybe implementing some of that into your standard operating procedures would be critical. Diagrams are helpful. They're really helpful because while you can practice this, if folks get insecure, they can always open an SOP, they can look to a diagram. There are great forward pipetting diagrams on the internet. They're all over the place. And they really help to visualize that. And yeah, definitely get folks to practice. And if you're still having problems, bring in an expert, bring in someone else, bring in an academic person, someone who can really help you hone these skills, because they're critical to the foundation of every microbiology lab.
Yes. Great, thank you. That was very thorough, and yeah, any skill you're trying to learn, the more you do it and the more you practice, and even within Virtuosi, you're able to practice those techniques over and over again in a risk-free environment, again, when no one's looking over your shoulder, you're getting cues from a mentor that is always happy to provide you feedback in the VR as well. So another opportunity there. Okay.
Oh, I just wanted to add one more thing, which is, sometimes you see this too. Obviously, everything that Vanessa talked about, make sure that you think carefully about what your analyst qualification program is. And I'm assuming that everything is properly calibrated and we don't have any equipment issues. But something else just to think, to keep in mind, because sometimes you do see it, is if every pipette that you use is from a different manufacturer, right, and they all have a different feel, even if they're all perfectly calibrated and the analysts are well qualified, every time you pick up a different micropipette, it feels differently, the stops are at a different location. That can really trip up even an experienced analyst. Just they can't get in the groove of having that feel every time they pick up a different pipette. It feels differently. So that's also just something to think about, is consistency of the equipment that analysts might be using throughout a test method or a series of test methods.
Yeah,
thanks
Tyler,
for
you bring
adding
up a
that.
good point. I've been in labs where I see, pipette aids labeled with initials, because microbiologists have like favorites, right? And that's because the lab had many different vendors. And they knew one worked better than the other, or one was more precise, or one had like a sticky trigger, finger button or something. And so it's a really good point, and it's not to be, sort of taken lightly. To understand calibration, replacement of any discs and stuff like that is critical. But also, yeah, don't have like 85 different options. Have the ones you need, have those appropriately calibrated, and then have everyone being able to feel comfortable with all of the different options, right? You shouldn't have that only one pipette that you like to work with. You should be more flexible and be able to use multiple types of equipment. So really great point, Tyler.
Yeah, this is great advice. Thanks, guys. Okay, moving on to a different topic here. What is the minimum temperature and duration required to get 3-log reduction of BET? We put you on the spot there.
So the first thing that I would say is validation.
Yeah, and I'm not sure. So for gel clot method, we usually do 37 degrees plus or minus, a half a degree.
Oh, no. This is incubation. This is for, I think we're talking about 3-log. So this is probably a depyrogenation process, would be my guess, if we're talking about.
Oh, why did I hear incubation? Oh, so for depyrogenation? Yeah, I'm not an expert on the depyrogenation process and the qualification for that.
Generally the minimum is 250 degrees Celsius and 30 minutes. But this is exceptionally important to qualify and so forth for whatever you're using, whatever your parameters are. This isn't a situation where there's just sort of a minimum that you want to be sure to meet. It's super important to make sure that this is properly qualified for whatever your particular product or process is.
Okay, and if that didn't answer the question, you can always contact us and provide additional detail for what you're looking for the individual who asked that question. And I'll give you contact information here in a little bit if, like I said, if that what did not answer your question.
Yeah,
okay.
We've got experts on our team who can answer that better, and we've got entire episode content on depyrogenation and the process for it.
Yeah, absolutely. Okay, with the expected this is interesting. With the expected increase in endotoxin testing as new vaccines become available, which method, kinetic or gel clot, is more likely to be employed by the industry at large going forward? And then the follow-up is, what are the benefits of a kinetic method over gel clot, if any? Sounds like they're asking to make a prediction here.
Yeah, so for me, I think kinetic is going to be probably faster. I think you can generally process more samples using the kinetic methods. They, typically take, 96 well samples, so you can process more. With gel clot, you have to consider how you're batching the samples, and you can't do too many, because if you're placing, let's say 96 samples in an incubator for gel clot, there's no way you're going to be able to thoughtfully invert one reaction tube 96 times in under 3 to 4 minutes, which is typically the time period to which you need to interpret those results. So I think, if I remember that question correctly, in general, kinetic is more likely to be employed by the industry because of its automation, because of its high throughput, because of also its quantitative accuracy. And I think that those are some of the benefits for the kinetic method. And there's even cartridge technology that employs the kinetic method. And so you can use even less quantity. I think it's like 1/20th of the quantity we require for lysate, and process more samples. So a lot of those efficiencies are already employed at so many different companies throughout the world and in the United States. And so it depends on the methods that are qualified and validated for the specific vaccines. If we're talking about COVID-19, then they would have to run, the method suitability and qualify that method. But it just, from basic understanding, it would be kinetic for me for a variety of reasons. What about you, Tyler? Any thoughts?
Yeah, and I think it's also important to mention that not all automated methods are necessarily kinetic. So it's sort of gel clot in one category, or a chromogenic, turbidimetric, and some of them are endpoint methods where we look at a particular time, whether a turbidity or a color has developed, or where kinetic method means that we're actually looking at how these things change over time. And at the end of the day, our result is the same. Is this, within specification or not? But the way the calculation happens is a little bit different. So I wouldn't want to say, no, it's kinetic versus gel clot, but sort of these more automated methods, and there are a variety of ways. But yeah, exactly. Like, I would agree with everything that Vanessa said. Sensitivity is better with these automated methods, and some of them, depending on all of the specifics of the reagents that are being used and the specific technology, are sometimes much less susceptible to interference, to enhancement or inhibition, which can be a problem with the gel clot. And also, I think something that Vanessa, just touched on, but the fact that these automated methods, have the data can be in a computerized digital format. Which can be really great, for reporting and integrity reasons and so forth, as opposed to gel clot method, which is, going to be recorded by an analyst. Having a digital record of everything that's done can feed right into a quality system depending on how everything's set up. So that can be a big plus too.
Okay, great. Thanks for all that information. Super helpful. So we're running close to the top of the hour, and we have so many more great questions here, but I just wanted to make a couple of quick announcements. For those who can only stay on for the hour, we absolutely appreciate your time. We know that you may not have been able to stay on for the entire time. So let me just make a few announcements and then we'll continue on with the Q&A session. So as I mentioned earlier, today is the third of 4 installments of Thought Leader Thursday. We have one more coming up next week, which is November 19th, and we're going to be talking about manufacturing equipment use cycle. So that'll be the last webinar, Thought Leader Thursday, in this series. So be sure to get registered. You can find us on LinkedIn. If you get an invite through your email, just make sure you get registered for that last session. And we've had such a fantastic response to our Thought Leader Thursday webinars. We are planning to continue them in 2021, so keep an eye out for announcements for our next installment of Thought Leader Thursday. And one last announcement before we get back to the Q&A with Vanessa and Tyler here. As I mentioned earlier, we're excited to introduce Takati, which is, really the next evolution in education brought to you by Quality Executive Partners, which is going to be launching in 2021. A couple of details about Takati. Takati offers an advanced level of learning experiences for site and corporate leadership and includes interactive lessons, discussions, and workshops with SMEs and peers to provide deployable practical knowledge. So the first session that we're going to hold for Takati is the Advanced Forensics Auditing for Data Integrity, and that one is scheduled for February 1st through the 4th. Our executive leadership team, Crystal Mersch and Brian Duncan, are going to be leading that session. There's going to be a ton of fantastic information. Each session is limited to just 48 participants, so you're going to want to sign up early. Registration and more information will be forthcoming, but you can check us out on LinkedIn at Takati is through LinkedIn, and you can also email us at takati@qualityexecutivepartners if you have any questions on that topic. So, okay, announcements over. We can get back to the fun stuff that you guys are all here, the question and answers with Vanessa and Tyler. So let's get back to it. Let me get the questions here. All right. If the incoming vials are tested for BET and nothing was found, should we worry about depyrogenation?
Interesting question. Tyler, can I take this one?
Oh yeah, please.
Yeah. So I'm assuming that these are glass vials for, an injectable product, which is why you're testing them for BET. There are a couple of things. One, the vendor that supplies your vials typically tests them for BET as well on their side before release. They would release that with a certificate of analysis for that lot that was manufactured for those vials, for those glass vials. That process that they use to depyrogenate them, I'm assuming that it's not done in-house, right? An incoming lot. To depyrogenate them should be qualified, right? And that qualification should be reviewed by your company. And that's part of the supplier quality agreement, is that you understand what methods they're using to depyrogenate your materials, what methods and what are the appropriate limits or specifications that they put on testing prior to release of that batch of glass vials to your company. And then upon incoming receipt of that batch, you guys are testing them. So I'm not sure what you mean as far as depyrogenation, but the handling and the incoming receipt and how those vials are loaded onto the manufacturing line all must be considered, right? So that you don't have any introduction of contamination in that process. And typically, the microbiology lab will do that sort of spot check, but understand that you have a qualified process to depyrogenate, that the vendor is testing them every lot, or they should be, and that all should be captured and reviewed in your supplier quality agreement. If they have any investigations on their end, you should know that prior to either receiving them or, definitely prior to using them on your manufacturing line. So I don't know what it means when you say, should we worry about depyrogenation? But if the process is qualified, you've got that supplier quality agreement, you're testing them, they're testing them, and the handling and placing of those vials on the line is all according to a qualified process. I don't think worrying about depyrogenation is something you have to do anymore because you've got all those controls already in place. So hopefully that answers your question.
Anything to add there, Tyler?
Only that just Vanessa is such a pro. It blows me away.
She has all the answers.
No, I do not. And it's hard, right? You want to make sure with the question that you're answering how it's intended. And sometimes we don't always know the nuance or the spirit of the question, but we definitely try our best.
Yeah, absolutely. And speaking of questions, if you haven't asked a question and you have one, now's your opportunity. We have about another 30 minutes in front of these two experts that will answer any questions on BET. So now's your chance. Submit those questions. So this is an interesting one that came in, and this is a little less about BET, but it says, how would we incorporate VR in BET testing? What would that look like? So I think I have some thoughts, not being a microbiologist, but being on the implementation side. But I'm curious to hear from Tyler and Vanessa what your experience is with the training using VR.
Yeah, I mean, first, if you're already having that question, you're in a good space, right? Because I think while you do get pipetting tips and all of that from academia, a lot of those skills that we were talking about earlier aren't necessarily something you're able to hone and practice when you get a job, right, in pharma microbiology for a company. So this changing climate, this changing economic climate, and the pool of candidates that are coming into microbiology labs are coming from all different disciplines, different walks of life. They can be from biology or chemistry majors. They can be from other sort of science majors. They can be pharmacists. They can have a whole different host of backgrounds. And I think that it's important as a company that you sort of standardize the expectations for your lab. And you standardize through a strong qualification program what it is that you want. And VR can absolutely be a really valuable tool to help you do that. So you have foundational education knowledge, and then prior to even doing a method, any method in microbiology, not just BET, you can practice that in VR. And we've seen the aha moment with microbiologists in VR when they're connecting, the content that they see in VR to the methods that they're doing in VR, again, in that safe space. You feel like, oh, we've seen at conferences, we've seen during user testing, we've seen during, deployments at different companies and sites, that aha moment. And that's the power of VR, amongst many other things. But the fact that you're including VR into your thought processes for those techniques, I think is great. This new generation of talent coming from college is thinking and learning in a different way, in a whole different way. And even regardless of that, the talent that's been on the market for 10, 20 years, they can use a refreshing, new, practical way to learn information, to learn skills. You know what I mean? I worked with a colleague, Jose, such a fabulous human being. He was a theater major, and he was in theater in San Francisco for like 20 years of his life. He went back to school, got a degree in microbiology, and came into the microbiology lab at age 50, full of life and spirit and zest to learn about microbiology. And he went into theater always knowing he wanted to go in science. And to me, that's fascinating. And if I had VR to help him understand, it would bridge that gap between what you learn in a book and when you have to go live on product samples. And that's what VR does for you, and it's really, really powerful. So I love that you're thinking about VR. I think you can contact Beth. She has all the solutions for you. And I know, Beth, you work with clients more directly than we do. We're on the technical development side of the product. And what kind of feedback do you hear from folks?
Yeah, so I'm so happy that someone asked that question, because these are the kinds of questions we actually get from a lot of clients, is how would we integrate this? What does this look like? So a couple of things that we've seen right off the bat. So first, we should say that, VR and the training within Virtuosi is not intended to replace SOPs. It's not intended to replace on-the-job training, but it bridges that gap, like Vanessa said. And we have done studies, I'll call them studies, but on different tenure levels of individuals within an organization. So initially, people's thoughts are, oh, well, new employees, this would be great for new employees because, like you said, Vanessa, they come from all walks of life. They may come from different industries. This would be good for new employees. But what we have found is that all the way up to 10, 20 years of experience, individuals are learning by going through the content within Virtuosi, because for a lot of different reasons, really, but some of it is someone taught them how to do something 10 years ago, and that's always how they've done it. They just don't even know why they're doing it that way. Or they were taught improperly, and that's just been carried through. And as that person has trained other people, it gets shifted, right, throughout the years. And now all of a sudden you're doing something, you don't know why you're doing it, and you're doing it maybe in an improper manner. So we've seen fantastic results with all levels within an organization and all years of experience. Another thing I wanted to mention is SME time. Something that organizations are trying to minimize is pulling SMEs out of their jobs. I mean, we all know, and Tyler and Vanessa, you are SMEs, you know that you are called on and pulled out constantly, because you're the people that we go to when we have questions. We find the most knowledgeable person, we find the subject matter expert, and we pull them out, and you're stretched way too thin, and the organizations can't support different activities if they don't have an SME available. So this, by utilizing VR, you can minimize some of the time that's needed to be spent on training someone to do pipetting, for instance, or the BET testing techniques. And we've seen that that's helped immensely within organizations. So I'm not sure that we answered the question specifically. There's lots of different ways to do it, and lots of benefits that we're seeing. So if you have any other questions, or that didn't specifically answer the question that you had, just I'll give you contact information on how to get a hold of us to ask that question. But I agree with you, Vanessa. If you're already thinking about how VR could work into things, you're on the right track. Tyler, was there anything that you wanted to add?
Yeah, Tyler, you're the expert on adult education and innovation, right? Like, how do you see VR being a part of every company, every laboratory? Now I'm asking you the question, future training program, because we love it, right? But as developers of this product, we're sort of partial. How do you see it from the larger scale for companies to implement?
Right. Well, what I like to think about is that VR, I think, is really able to provide the training opportunities that we all wish we could, but we're often just unable. SMEs don't have enough time. Laboratory managers, more experienced analysts, don't have enough time. Like, ideally, how would we want to train folks? We'd want to be able to pair them up with a mentor who watches every move. The individual earlier had a question about pipetting errors, right? And it really comes down to analyst qualification. But think about what a really good qualification system involves, and there may be folks who really need to practice, and there often just isn't time for managers, for more experienced analysts to watch them and say, oh, do this a little bit differently. But that's the most meaningful training. And what's so cool is how VR can do that, right? VR can really be that constantly vigilant mentor who's watching the learner from all angles. And, oh no, remember to eject the tip like this. Oh, remember to go to the second stop or the first stop here, and so forth. And it's so cool, because I think we all learn best by trying stuff over and over, getting, helpful patient coaching. Nobody likes being screamed at by somebody who's, at the end of their rope, and I've watched you do this all day long, and just why won't you get it? VR doesn't do that. VR is like, oh, maybe remember to go to the second stop on the pipette here, which is great because it's everybody learns at different rates. So that's what I always say. I say, imagine the best program that really patient managers and SMEs would put together, constantly monitoring your folks, constantly mentoring them. And that's what VR can provide, and giving people this holistic, hands-on sense of how what they're doing in the lab connects to the theoretical and what the best practices of the day-to-day should look like. That's what's so exciting for me about it.
Yeah, I'll just add one more thing and we can move on. But something that you said, Tyler, that I hear all the time in the field is that SMEs or trainers have, sometimes 6, 8, 10 people they're training at one time. And this maybe goes beyond BET testing, it just generally they have a bunch of people they're trying to train, and you clearly can't have eyes on everybody at once. So, someone makes a mistake, you don't see it, that person doesn't know they made the mistake, they move on, and either that mistake is caught later, or worse, it's not caught, and now we've got an investigation going on for some particular purpose, and you identify it hours and hours and dollars later of investigating something that happened. So that's definitely a benefit that we've seen just being out in the field, that folks are benefiting from the VR. So
yeah, I think it's 2020 and there's a lot of technology out there that can help us perform better, be more efficient, and take a lot of those pressures, right? So in 2020, manufacturing hasn't slowed down. It's gone faster. More companies are popping up, more projects are popping up. Now we need extra billions of doses for a global pandemic, right? Everything's going faster. And I think if we were to support our training programs with the technology that's available, we should. We absolutely should. It's that simple. So this is a great tool for all folks to consider implementing.
Yeah, absolutely. Okay, next question. A lot of times we review BET out of limited investigations that have an automatic hypothesis retest with multiple replicates/analysts to determine lab error due to inherent contamination risk of the test. Is that a typical practice in industry? And if yes, what are some of the reasons for the inherent contamination risk?
Oof. So I'm hearing a lot of terms that just in the root of the question concern me, right? So automatic hypotheses, inherent contamination risk. So I don't know what those mean. Inherent contamination risk isn't something for bacterial endotoxin that we want to consider in routine. You consider what are the contamination risks to your method and to your process all the way up front. This needs to be a forward-thinking approach, right? You can't in routine assume there's inherent contamination risk. You have to remove that, and we talked about that earlier in the webinar. You have to remove those potential risks for contamination of your supplies, of your analysts if they've got poor technique, or other sort of plastics. If you're parafilming a reaction tube, that's going to be a potential source of contamination, something like that. So during method suitability, you have to use qualified reagents, right? Appropriate qualified reagents that the industry use. You have to use materials that are pyrogen-free or depyrogenated, and that they're handled appropriately. You've got to use qualified people who have the appropriate pipetting techniques, the appropriate contamination prevention aseptic techniques during that. And all of this matters because you pull that contamination risk out of your process and out of your method in routine. So if you are automatically assuming that, I think you have to consider how you're executing your method suitability program in the first place, okay? The fact that in the question, it was automatic hypothesis for retesting. So I think that the spirit of that comes from a place of maybe not having the full knowledge in understanding how investigations should be structured in microbiology. Typically for BET, we talk about phase 1 and phase 2 investigations. Phase 1 focuses on the laboratory and whether or not you have a valid test, okay? And invalid tests are those where the system suitability parameters that I talked about for the method, such as negative controls, positive product controls, confirmation of the label claim if you have a gel clot technique, generating a linear standard curve, right? If these things don't function as expected, as you've proven in your method suitability, then they may affect the accuracy of your test results. And that's what we would call an invalid test. Only if you can prove during your phase 1 laboratory investigation that you had an invalid test, can you then consider whether or not retesting is appropriate, depending on which portion of those suitability parameters was invalidated, and how you would go about retesting and whether or not it's appropriate. That retesting hypothesis needs to be approved by management and QA. The very nature of that exercise means that it's not automatic. This should be rare, right? You should have considered all the potential interference of your product, the potential contamination risks up front, and built a method suitability program that makes sense for routine testing. So I'm basically offering advice for you to go back to the fundamentals of your method suitability program, to understand where those sources of contamination could come from and how to remove them so that you don't have to experience this in routine. You shouldn't be experiencing or concerned about contamination risk in your test method. And you should be more confident in that your program's built, and I get that things happen. So that's what the phase 1 laboratory investigation will determine, that something went wrong, that an SOP a step was missed, or that the wrong reagent was used, or that a reagent was expired, or it was, some contaminated because it was past its 24-hour expiry. Whatever it is, you do that root cause analysis in your investigation. If you find that nothing could be invalidated from the laboratory perspective, then you move to a phase 2 manufacturing investigation and determine if contamination of endotoxin genuinely happened in the production environment. And that could come from a host of things. But manufacturing has already considered what are the potential contamination sources in our process and how to remove them as well. So maybe a vendor for a raw material changed, and their limits for endotoxin are less stringent than your prior vendor, who knows? Maybe something went wrong with depyrogenation. Maybe the cycle failed when you were depyrogenating, your vials, if you do that in-house. Maybe you had a leak in, the manufacturing space where you filled product, and that could be a source of gram-negative bacteria. Who knows? But the laboratory can't automatically assume and automatically jump to a hypothesis of retesting without probable cause that relates to whether or not validity was obtained during your test method. So I caution you to go back and think on the basics of the suitability program, and work with your colleagues to maybe restructure that. And I caution you not to have anything automatic about retesting. Okay. Sorry, that was a long-winded answer, but it's a very important topic. Investigation of results. Oftentimes the lab gets a lot of blame and pressure to just retest, and you cannot automatically retest. That would be retesting into compliance. And using, different analysts and different equipment and stuff like that can happen if you've got a real reason for an invalid assay, and you've proven that, and you've got a hypothesis that makes sense, and that's approved by QA. So there's a lot of structure that needs to go into this type of investigation.
Yeah, that's thanks for that, Vanessa. It sounds like there's all sorts of things that could go wrong, and never making the assumption. Okay, next question. Sir and ma'am, please tell me what we must know for, and I'm probably going to botch this here, so you guys can correct me. RFC testing, is there another way to say that?
Yeah, so that refers to recombinant factor C, which is that sort of laboratory produced synthetic version of the key component that is in LAL.
Okay,
and so
great.
what was the full question that they'd asked?
Yeah, what do we need to know for that kind of testing, for our recombinant factor C testing?
Right. Okay. Well, there's a lot that you'd need to know. You know, without the full picture, you know, it's hard to give all the specifics. But I can talk broadly about it very quickly. You know, the idea is, as we mentioned earlier, is that recombinant factor C is a synthetic version of the key component of LAL. It's the protein that detects the endotoxin and then starts the sort of cascade of clotting. And in tests that use it, it's usually not causing clotting. It's usually causing the cleavage of a molecule that turns a color after that cleavage happens. So we tend to use it in chromogenic endpoint tests. Now, the thing that's important to mention, sort of as we've talked about earlier, is this has not been a fast transition in the industry to using recombinant factor C. This has gone a little bit faster in Europe with the European Pharmacopoeia than it has here in the US. So this is using RFC is considered well with the new general chapter of the EP coming out at the beginning of 2021. There'll be a general chapter devoted just to RFC testing. And, you know, it can be considered a compendial method. In the US, for the USP, it's been much slower. The new chapter 1085 that talks about recombinant factor C is an informational chapter or one for guidance. And so using it is not considered a compendial method, which means that if you're planning to use recombinant factor C, you'll have to go through the validation protocol that you would, 1225, 1223, for using an alternative method for endotoxin testing. So that's, at the big picture is, as I like to say, validate, validate, validate. And without a whole lot more specifics, there may be a particular thing that this individual is curious about, but I think that's sort of the big picture, that it's not fully globally recognized yet as a compendial test method. So you look at guidance carefully.
Yeah, and I think if I may add a less nerdy response, as Tyler, because he's always teaching me the science of these things, right? And so if I were to think about the compliance aspect, I mean, in microbiology, it's really difficult to inspire change. And part of the apprehension with adoption of something like recombinant factor C is rooted in that apprehension for change. And I think that we haven't yet, as an industry and as a collective microbiology laboratory, proven that, demonstrated track record for validating this method enough. And we've got years, like tens of 20s of 30, 40, 50, 70, 80 years, of validation with the Pyrogen rapid, the rabbit Pyrogen test. And then we moved over to, the way that we use lysate now from natural sources to indicate presence or absence of endotoxin. And then now we have to make, as an industry, that shift towards an alternative, recombinant factor C. And I don't think the idea is that RFC is going to totally replace. I just think that it's another tool in the toolkit of a microbiology laboratory for how we can test this, right? Without that reliance on horseshoe crabs. And I think that it's a very, like Tyler alluded to early, controversial topic, because horseshoe crabs, it's not just about them. They're part of this ecosystem, right? Especially on the Atlantic coast. And it's important to understand that, you know, for a whole industry to be dependent on one species of, you know, animal is something that we don't have to be. There could be other options. It's not one or the other, it could be a combination. So getting folks to adopt recombinant factor C at a company level and getting them to do the alternative method validation with that side-by-side comparison, more folks to do that in time will then bring on the adoption of this new test method. And then I think we parallel this same concept with rapid technology. The technology is there, the science is outstanding, but we still haven't had a lot of folks shift to rapid technology because we don't have enough companies employing the method, doing the validation, and having the data in routine to support, like, hey, yeah, this is no problem, we've got to move towards this. So we tend to be a little bit more apprehensive in microbiology, and I think that, you know, with the adoption of new test methods, new technology, some companies just got to go for it, right?
Yeah. Yeah, that sounds great. Thank you so much, Tyler and Vanessa. So we're running close on time here. So I think that's the last question that we're going to take this morning. If you had a question and it did not get answered, you can email us at info@virtuosivr.com. So again, that's info@virtuosivr.com. If your question didn't get answered, or if you think of a question later that you would have liked to ask, go ahead and send that our way. So Tyler, Vanessa, thank you so much for your time today. I learned so much, and I know that the audience did too. And I'm looking forward to next week. So next week is going to be the fourth and final installment of this series of Thought Leader Thursday. On November 19th, we'll be covering manufacturing equipment use cycle. So keep an eye out for an invitation to register, and you can find us on LinkedIn and register that way as well. So just another couple of quick announcements here. We will be holding Thought Leader Thursday webinars in 2021. So keep an eye out for that. And yes, it's a huge hit. We have such a fantastic response, and we want to keep doing this. We want to keep providing, you know, information from our subject matter experts like Tyler and Vanessa to help educate and inform the industry. So one last announcement, I wouldn't want to leave us without another just shout out to Takati. So if you or someone you know is interested in experiencing the next evolution in training, follow Takati on LinkedIn. All of the updates will be happening there. You'll get firsthand information on the Takati program. So there are going to be a number of exciting programs in 21 related to Takati. So just as a reminder, Takati offers an advanced level of learning experiences for site and corporate leadership on complex issues facing the entire industry. So it includes interactive lessons, discussions, and workshops with SMEs and peers to provide deployable practical knowledge. Again, the first session is going to be on advanced forensics auditing for data integrity, and that one is scheduled for February 1st through the 4th. And members of the Quality Executive Partners leadership team, Crystal Mersch and Brian Duncan, are going to be leading that session. And again, the sessions are limited to 48 participants, so you're going to want to sign up early. Registration and more information is coming soon. You can follow us on LinkedIn at Takati. So thank you so much for your time today, everybody. I hope you enjoyed this as much as I had, and we look forward to seeing you next week.
Yeah, thanks to everyone who helped put us together for this webinar today. It's really a great effort by a huge team. It's not just us. So thanks to everyone for attending and all our microbiologists out there. We'll catch you on the next webinar.
Sounds great. Thanks, everybody.
Good. See ya.
Bye, Tyler. Bye, Beth.